present our recent work on applying an elastic network model to study the chromosomal dynamics. Fortunately, this work has led to a publication, and this is actually the full citation to the paper. And if you are interested after this talk, please feel free to check out the paper. So, elastic network models were introduced in the field of protein dynamics 2 decades ago for characterizing large-scale motions of proteins. The protein molecule is first converted into a network representation, specifically a Gaussian matrix. And by solving this rather simplified system, we can obtain modes of motions that dictates the overall dynamics of the protein. And using these modes, we can compute a lot of dynamical property of the structure, and for which I will talk later. With the data from the contact data from high-C experiments, we can also apply the similar model to analyze the chromatin dynamics as well. So the high-C matrix itself is already a network representation of the chromatin topology, where the loci are the nodes and the interaction frequencies are the edges. Without the 3D information, it's impossible to know the exact motion in 3D space, but we can still use this model to compute a lot of dynamic properties, including the mobility profiles and also the covariance measurements. So in this plot, the blue curve shows the mobility profile predicted by the GNM for the loci on chromosome 17. And we found it's highly correlated with 2 independent chromatin accessibility datasets, ATTAC and the DNA-seq. This correlation makes sense because if a region is exposed to the environment and therefore accessible by the enzymes, then it should move more freely than the regions buried inside, as illustrated by this figure. And we found it is true. The correlation is true for all the 23 human chromosomes. Another important question in many chromosome studies is to understand the long-range interactions. So the covariance matrix evaluated from the GNM can show how much 2 loci are correlated or coupled in terms of their dynamics. The coupling may be a result of either direct or indirect contacts, and the latter of which may not be captured by the high-C experiments. If you look at the covariance map, you can see this green off-diagonal block, which is what we call cross-correlated distal domains, or CCDDs. They correspond to the 2 domains that have long-range interactions. And these long-range interactions, you can identify them from the covariance matrix very easily and systematically. And in our paper, we showed that the gene pairs that are located in the CCDDs actually have higher co-expression values than the gene pairs located outside of the region. And as I mentioned, the modes of the motions can be given by the network models. And in GNM, they are in one-dimensional. So it's impossible to know the exact motion of the molecule. But still, the modes can be used to identify the chromosomal domains by simply finding all these crossover sites in different modes. In protein analysis, these sites are referred to as the hinge regions. And by using different number of modes, we can identify the domains at different scales. We compared our GNM domains with 2 known structures. Firstly, the compartments and then the TADs. We found out that using fewer number of modes, which is 12 in this case, our domain look very similar to the compartments. And if we add more modes, if we use 35 modes, around 35 modes, our domain started to look smaller and finer, and they will be more similar to the tags. Okay, and that, in summary, the only input for the Gaussian network model is the high-C matrix. And what you can get is the mobility predictions, which is shown to be correlated with the chromatin accessibility, and also the covariance matrix for identifying long-range interactions. And then you can also obtain the domain separations at different scales. And I would like to mention the runtime of this algorithm of the model is really fast. It's around 1 minute per chromosome for 50 KB resolution data. And it will take longer time for a larger data set, of course, but this might be some thing you can check quickly whenever you get a new data set. And I would like to thank our collaborators from CMU, especially Natalie, who is the first co-author of this paper and helped a lot with this talk. Thank you very much.